So I started this class by discussing, or this session by discussing the RNA structure. And we know what is RNA, and the explanation we gave in the formation of a polynucleotide. We said in the case of RNA, what makes it different from that of DNA is that in the case of RNA, we have a polymer of ribonucleotide instead of the polymer of deoxyribonucleotide. So RNA exists not as a double strand, rather a single strand. But the RNA single strand can sometimes form a secondary structure. How? RNA can fall back on itself to produce a different form of structure where we mention loop, hairpin, stem loop, etc. The image here by your right is an example. So we said we have a different type of RNA, several types of RNA, but the most common ones are the three types of RNA. Number one, we said we have the messenger RNA. Remember, we said the genetic material in DNA contains genetic information. Now, if this genetic information is going to be utilized, most especially to produce a protein, that genetic information is copied to RNA. The type of RNA that contains this genetic information is called the messenger. So the messenger RNA is the one that is involved in the transfer of this genetic information, usually from DNA, and then it is utilized to produce a protein. Is that clear?
Yes, sir.
Another type of RNA is the transfer RNA. The transfer RNA is the type of RNA that is involved in the transfer of amino acids during protein transfer. So when you have a messenger RNA that contains an information for a particular protein to be synthesized, this messenger RNA will attach to a machinery, something like a machine. And that machine will read the messenger RNA. I will tell you which machine is that shortly. When the messenger RNA attaches itself to a particular machine, that machine will now read the messenger RNA, the information on the messenger RNA, and say, oh, this messenger RNA says we are to produce a protein. And that protein contains so many amino acids. Number one amino acid, let's say, is arginine. So number one amino acid is methionine. So where do we get the methionine? So transfer RNA will now bring transfer methionine. Messenger RNA contains the information, and that information will now be read by a machine. I'll tell you which machine is that. That machine will now say, oh, to produce a protein from the information here on the messenger RNA, we need some amino acids. Number one amino acid, for example, it will read methionine. Okay, where do we get the methionine? The transfer RNA now is responsible in bringing methionine. So transfer RNA will transfer the amino acid, methionine. So when methionine is available, okay, now it will release methionine. Then the next amino acid, the next machine will now read, the next amino acid is arginine. So another transfer RNA will now bring arginine again. The next one is glycine. Now another transfer RNA will bring glycine. The next one is going to be serine. Now another transfer RNA will bring serine. Did you understand? Did you?
No, sir. So the messenger, is it the same thing with the messenger RNA and the transfer RNA?
Listen, please. Messenger RNA is the type of RNA that contains the genetic information. Remember, we say DNA contains the genetic information, right?
Yes, sir.
And most of those informations in the DNA, they are used either to produce a protein or sometimes to produce the RNAs, because the RNAs are many. So if the information needed, for example, is to produce a protein, now this information will be copied into RNA, and that RNA is called the messenger RNA.
Okay.
So any information that is needed to produce a protein will be copied from DNA to a messenger RNA. So messenger RNA contains that information that will be used to produce protein. Is that clear? Is that clear?
Yes.
Yes, sir.
That is messenger RNA. Now the next one I said transfer
Yes,
RNA.
sir. It is.
Now transfer RNA, what does transfer RNA do? Transfer RNA, it transfer amino acids during protein synthesis. It bring amino acid. So now I'm telling you, I'm trying to link the function between the messenger RNA and the transfer RNA. So when the messenger RNA containing the information needed for protein synthesis, that messenger RNA will now travel and then attach itself to a particular machine. I'll tell you which machine is that. It will attach itself to a particular machine. And that machine is the one that will read the information on the messenger RNA, because the messenger RNA contains information needed for protein synthesis. So when it reads the information, it will read, okay, we need to synthesize the protein. And this protein, the first amino acid on this protein is methionine. It will read methionine. After reading the methionine, now a message, a transfer RNA will now bring methionine, because its work is to transfer amino acid. Now, after it brings methionine, now the messenger, the next machine will now read which amino acid is after methionine. After methionine is serine. For example, I'm giving an example. Now, the transfer RNA will go and bring serine. After serine, the machine will read, okay, the next amino acid is valine. So the messenger, the transfer RNA will bring valine. Next one is tyrosine. The transfer RNA will bring tyrosine, like this, like this. Do you understand?
Yes, sir.
Do you understand?
Yes, sir.
Now, the next type of RNA is the ribosomal RNA. Ribosomal RNA.
Take it again.
Ribosomal RNA is written here. The first one is the messenger RNA, which I explained. After it, you can see the third one is the transfer RNA, which I just explained. The next one is the one after transfer RNA is ribosomal RNA. So this ribosomal RNA is the one I call machine. It's the machine. It provides a structural framework for ribosomes. So this ribosomal RNA is usually what we see as ribosome. We know ribosome, right? In the cells.
Yes.
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 So
Yes, sir.
basically contain ribosomal RNA. And that ribosomal RNA is the machine where the protein synthesis occurs. It is where the messenger RNA will go and attach itself. When it attaches itself, the ribosomal RNA will now read the messenger RNA. With the messenger RNA, it will know the amino acids that are needed. When it reads the first amino acid, say methionine, now the transfer RNA will bring methionine. It will go to the next one, serine. Transfer RNA will now transfer or will bring serine, like this, until when you have a long peptide, a polypeptide. That polypeptide can be folded to form protein, which we already explained in our previous classes. So basically, these are the three main types of, or the major types of RNA: the messenger RNA, the transfer RNA, and ribosomal RNA. But there are other types of RNAs. There are other types of RNAs apart from these three major types. For example, we have heterogeneous RNA. Heterogeneous RNA is like a precursor of messenger RNA. When messenger RNA is going to be produced from a DNA, supposed to only produce RNA, and that RNA contains some sequence that are not needed. So this sequence will have to be removed. After removing this nucleotide sequence, the remaining will be joined to form the messenger RNA. So the first RNA that is being produced is not the messenger RNA from DNA, rather it is heterogeneous RNA. Now, after heterogeneous RNA will undergo some processing, some things will be removed from heterogeneous RNA, some nucleotide sequence will be removed. When they are removed, there are some particular type of RNAs also that also do that, removing some nucleotide sequence. Then the remaining one will be joined to form the messenger RNA. The next one, we have small nuclear RNA. Small nuclear RNA is involved in the processing of messenger RNA. We also have small nucleolar RNA, which also involved in the processing of ribosomal RNA. There is also small cytoplasmic RNA that is involved in the selection of protein for export. When the protein is produced, it needs to be taken out to a different organelle. Then this protein will be the one involved in the selection of which protein to be taken out, which ones is not going to be taken out. There are also quite a number of them, for example, that are involved in the regulation of gene expression. We have microRNA, we have small interparent RNA, etc., etc. There are many. You can, on your own, you can read about them. Now, difference between DNA and RNA. I think we have already talked about difference between DNA and RNA with respect to the bases. We say that DNA contains all the bases with the exception of uracil, right? While RNA contains all the bases with the exception of thymine, right? While with respect to sugar, we said DNA contains the oxyribose sugar, while RNA contains the ribose sugar, right? Class, is that true?
Yes, sir. Yes, sir. Yes, sir.
Now, with respect to structural conformation, we said, or we just mentioned that DNA exists in double strand, while RNA exists in single strand. While with respect to function, we said DNA contains genetic information, while the function of RNA is versatile. We just mentioned that some RNA form a catalytic function like an enzyme, some RNA form like regulatory function, some RNA like messenger RNA, they are codings, they code for information also like DNA. Some RNA like transfer RNA are involved in the transfer of amino acids, etc., etc. So with respect to function, RNAs are versatile. With respect to stability, RNA tend to be less stable. Why they are less stable? Simply because there is an OH group attached to carbon 2. And that OH group has the potential to undergo a chemical reaction. Therefore, it is not stable. RNA can easily undergo reaction because of the presence of that OH group, while DNA does not have the OH group. Therefore, it cannot easily undergo reaction. Is that clear? Is that clear?
No, sir. Yes, sir. Yes, sir.
What is not clear? What is not clear?
So I see the difference between the DNA and the RNA.
Yeah, what exactly is not clear? What exactly is not clear?
The explanation of the explanation you are giving, sir.
Have you been part of this class, Blessing?
Yes, sir.
Because this thing I just mentioned is like a repetition of what we discussed in the previous class.
I've been part of it, just I was obstructed by network.
Okay. Now, the difference we mentioned are in 5 ways, although here we listed 4 ways. Number 1, the basis. RNA and DNA differ in bases. Even though RNA contains 4 bases, DNA contains 4 bases also, but RNA and DNA have 3 bases that are in common.
Okay.
While the 4th one, for example, in DNA we have thymine, while RNA does not have thymine. RNA have uracil, while DNA does not have uracil. So this is a difference based on the basis, the nitrogenous base.
Okay.
While based on the sugar, we know that DNA contains deoxyribose sugar, while RNA contains ribose sugar. And we said the difference between the deoxyribose and the ribose sugar is that in ribose sugar, there is an OH group attached to the carbon 2 of the ribose sugar, while on the deoxyribose, there is no OH. With respect to structural conformation, we said RNA exists as a single strand, while DNA exists as a double strand and helical structure.
Okay.
With respect to function, we say DNA contains genetic information.
Okay.
DNA contains genetic information, while RNA, with respect to function, is versatile. It has a lot of functions. Some of the functions we mentioned is that it is catalytic in nature. It can play a role like an enzyme. It can be regulated in nature. It can regulate chemical reactions, or it can regulate signaling pathways, etc. And at the same time, it can also code genetic information like the DNA, which we just mentioned, messenger RNA containing some genetic informations. Now, the last one, which was not mentioned here, is with respect to stability. Now, we said DNA tend to be more stable than RNA. And this is just an explanation we gave. What makes it less stable is that the OH group that is present at the carbon 2 of the ribose sugar in RNA, it is very reactive. So that reactivity can make the RNA to easily undergo reactions. That is why it is not stable. While RNA, being the fact that RNA does not have the OH group at carbon 2, making it to be less reactive, therefore less stable. Is that clear?
Yes, sir. Yes, sir. It's very clear.
Good. Now, I will come back to this. I will come back to this slide. I think we're supposed to explain this before that slide. I'm supposed to explain some things. So please pay attention to this, okay? Class, are we together?
Yes, sir. Yes, sir. We are.
Pay attention to this. Now I will talk about what we call a central dogma of molecular biology. Now, we said DNA contains genetic information, right?
Yes.
I'm coming. Let me stop the video and record again so that